We've finished with the first quarter of the semester, if you will, and what we've covered so far, I like to consider the macro part of microbiology. We've done a survey of the organisms that make up the microbial world, looking at the prokaryotes, the eukaryotes, and the viruses. And then we've also looked at their environments in general, the ecology of the microbes, how we've used them to help us treat water, and some of their properties with regards, their characteristics with regards to food. Now we're going to kind of shift our attention and we're going to move away from the macro aspect of it and look at the molecular aspect of microbiology. For the next couple of lectures, we're going to talk about the structure of the bacterial cell, of the prokaryote. We'll also compare that to the eukaryote. We're going to look at ways that we can control microbial growth. We can enhance microbial growth. Then we're going to look at the metabolism, what microbes use to grow with and some of the byproducts that they produce. We'll look at respiration, glycolysis, fermentation. And then we'll finish this section looking at genetics, bacterial genetics, and how we can manipulate that genome, if you will, to benefit us. So tonight, we're going to look at Chapter 3 in your book. We're going to skip right over Chapter 2. Chapter 2 discusses chemistry, and it was a prereq for you to get into this class to have had chemistry. And so we're just going to go right past that. If you need a refresher, I encourage you to just go back and read Chapter 2. We're going to move on to Chapter 3. Chapter 3 opens up with a discussion on microscopy. I'm not going to spend a lot of time tonight discussing it. We've discussed it a little bit in laboratory. We talked about light microscopes and what you have, a compound light microscope. The other microscope that I want to mention to you that your book discusses a little bit and actually provides us with some really neat pictures, if you will, is the electron microscope. The electron microscope uses electromagnetic lenses, creates a beam of electrons that it uses to bombard the sample, and then this is going to generate an image for us. There are two types of electron microscopes. There's a transmission electron microscope, which is kind of what I was describing just now. It uses a tungsten filament to generate a beam of electrons, and then it bombards your specimen with this beam of electrons, and on the other side is a fluorescent screen. Those electrons that hit something get deflected, and so they don't hit the screen, and so what you get really is a negative image of the specimen that you're looking at on the fluorescent screen. The great thing about transmission electron microscopy is that it allows us to see things with great resolution. Not only does it increase the magnification some 100,000 times, but the resolution is down to 0.3 nanometers. Now remember, resolution is the ability to distinguish two things as individual objects or points. When you get too far away from things, things start to fuzz together and you have difficulty distinguishing them apart. I can put two little dots here, and my guess is that you can't see them. Maybe you can if you've got really good eyesight, but they probably look like one point to you from a distance. It's not until you get close to it that you can see that you have two distinct points. That's what we mean by resolution. And the transmission electron microscopes allow us to resolve things to a distance of 0.3 nanometers. So it allows us to see things that are inside the cells, the molecules that make up the cells. We can look at the mitochondria, we can look at the chloroplasts, the organelles that are there. There is also a scanning electron microscopes. The scanning electron microscope creates again a beam of electrons, but this beam of electrons now scans over the specimen, and this specimen has been coated with a metal. A lot of times it's gold, and the electrons bounce back. And so you get an image that is going to be a three-dimensional image of the outside of the specimen. So transmission electron microscope gives you pictures of the inside of cells, if you will, because it's penetrating the cell, whereas a scanning electron microscope gives you a 3D image, a picture of the surface of what you might be looking at. Okay, so your book talks about some stains and techniques. I'll pass over those. We'll talk about staining when we get into laboratory. I want to move on to talk about the cell itself, the prokaryotic cell, the structure of the cell. When we talk about prokaryotic cells, bacteria generally, we can start by describing their morphology. You know, what do the cells look like? Well, there's a limit to the shape that these cells are going to be able to take. There's really only three main shapes that we can use to describe the cells. There are rods, which are cells that are longer than they are wide. Sometimes you see them referred to as bacilli. Then there are cells that are cocci. These cells then are spheres. And then you have cells that are spirillum. And these kind of corkscrew. Now you can have variations on these, where you can have some that are they're more kind of like a cocci, kind of like a rod. They refer to them as coccobacillus. You can have some that are curved rods, like this, that they would call vibrio. But generally, most of the time, you're going to be able to describe the cells based on these three shapes here. A more of a corkscrew type structure would be called a spirochete, but again, we can stick to these three main morphologies to describe most of the cells that we would come in contact with. There are a few cells that don't fit these descriptions, some of them that take irregular shapes. We'll always find exceptions to the rule here. The other thing that we can talk about with regards to morphology is how the cells cluster together. Now we know that when bacteria reproduce, they do so by a process called binary fission. Essentially, the bacteria just split into two. They increase in size, they replicate their genetic material, and then they split in two. Well, this can cause certain groupings to occur depending on the dividing and the process here. You can get chains that form where the cells don't separate you can get packets that occur where the divisions are perpendicular, and so you get very regular packets of cells. Tetrads, a lot of times cells will come in groups of 4, sometimes they'll come in groups of 2. And then you can also get clusters, where the division planes are random, and so what you see are groups of cells that look like bricks. You get these clusters there. So whenever you're looking at microbes under the microscope, two of the first things that you're going to note about them: what is their shape? What do the cells look like? rods, cocci. And then how are they arranged? What are their groupings? You know, they may be individually arranged, sure, or they may be in chains, packets, or clusters. So those are the two things that you'll want to observe about your cells under the microscope. Now let's look at the structure of the cells. And we'll start by describing the cell membrane. So I'm going to diagram a prokaryotic cell here, and this is the cell membrane of that cell. The cell membrane, the plasma lemma, the plasma membrane, the cytoplasmic membrane, all terms you might hear to describe it. This structure serves to regulate material in and out of the cell. It's the barrier. It really provides the cell with its integrity, controls what moves in and out. If you think about it, this is how the cell interfaces with its environment. Everything that the cell is going to encounter first encounters the cell membrane. So it's how the cell interfaces with its environment. And when we look at its structure, when we look at the cell membrane, we see that it's composed of a phospholipid bilayer. What that means is that we actually have two leaflets, if you will, that make up our membrane.
Is this layer composed of the fat?
No, this is the cell membrane.
The outer part?
Yeah, so we've got a phospholipid bilayer here, and it's composed of two of those phospholipid layers, and the phospholipid